She is in the Department of Neurosciences at the Mayo Clinic in Jacksonville. She describes herself as a translational neuropathologist who specializes in clinico-pathologic, genetic and neuroimaging aspects of aging and neurodegenerative diseases. Her expertise incorporates digital pathology. This is an ultra-resolution digital microscopy technique that quantitatively measures neuropathologic burden and cellular density. Her work is focused on a number of things, including clinical pathologic differentiation of atypical Alzheimer's disease, genetics of atypical Alzheimer's disease, neuroimaging of AD, and neuroimaging and neuropathology of dementia with Lewy bodies, as well as the neuropathology of tauopathies and TDP-43. She has received a number of honors, and among these are Alzheimer's Association de Lyon Prize in Neuroimaging, the International Franz Nissl-Young Investigator Award is granted by the Society of Neuropathology. And a while back, she was co-recipient of the Robert Terry Award, who is a very special person for us to remember. So today's presentation will be in a journal club format, as you know. This is a very recent paper just published in the last few months in Molecular Neurodegeneration. The name of the paper is Global Neuropathologic Severity of Alzheimer's Disease and Locus Coeruleus. Vulnerability influences plasma phosphorylated tau levels. Thank you again for being here, and I'll let you take it away.
Thank you very much for the opportunity. So I thought it would be nice to kind of extend some of what we talked about when I visited you guys in 2017, but more so transition to a whole new era of our research. My lab, as you so eloquently described, really capitalizes on digital pathology, meaning that we scan the microscope slides to allow us to quantify the brain changes. So whether that's lesions, from a density perspective of more of a burden analysis. So for the most part, we live in an insoluble world. We look under the microscope, we look at these lesions, or we work with neuroimaging, so structural MR or other types of tau or amyloid PET. But I had not had an opportunity to really work with fluids, and namely because the way that we've set it up is if our study participants have participated sufficiently until their untimely death, I'm able to really make those nice clinical pathologic comparisons. But really until recently, with the explosion in the field of fluid biomarkers, I hadn't had the luxury. And so this particularly is a study that gives us that opportunity to really kind of delve in and think about Alzheimer's disease from really a different perspective. But I thought I'd first introduce myself. So I co-direct our Mayo Clinic Brain Bank with Dr. Dennis Dixon, who actually was also co-trained by Dr. Larry Hansen, by Bob Terry. And so I was just talking with Christina earlier. We have a very similar sort of approach, diaflavin, to what was traditionally used as part of UCSD. And I also direct the Translational Neuropathology Lab, which is a fantastic group of humans. And our goal is to prevent Alzheimer's disease and enjoy the process. We really try to work together to think about things from the end point. So we begin at the end to inform the beginning. So we start under the microscope, and we always try to keep that human condition in mind. And really, we're dedicated to the study of two major topics. There's lots of overlap and there's lots of subtopics, but if we were to kind of capitalize or capture what we study as heterogeneity of Alzheimer's disease, whether that's from the age onset perspective or from the memory, non-memory, so non-amnestic forms of the disease, atypical Alzheimer's. We really focus a lot on neuropathologic underpinnings of Alzheimer's disease biomarkers. And for the most part, this was really in the neuroimaging sphere, but with the advent of the fluid biomarkers and the accessibility, this study specifically will evaluate the plasma biomarker. and focusing on the p-tau. Okay, so I really like to begin at the end to inform the beginning. So I'm going to start with our final figure and really kind of take you through the process. I thought this might be a neat opportunity to explore this in a journal club fashion, but with a little bit of a theme based. And so our theme is going to be mystery. And the mystery was, why does a plasma phosphorylated tau measure so strikingly associate with amyloid beta pathology in the brain? And this was a mystery that was brought to my attention through the literature, through people coming up to me, because for the longest, I guess for over a decade now, one of our sheer delights is to look at neuroimaging, understand the changes, and pinpoint the neuropathology that contributes to it. So we hadn't had that luxury with the fluid biomarkers, but this was our challenge. And so I'd like to introduce the two main characters, that being A beta plaque development in the insoluble form. So we look under the microscope. And then we also have neurofibrillary tangles. And I like the green. Oh, I don't like that green. And so what this will give us the opportunity to do is talk about the study through the context of both of these main characters. But I'd also like to introduce you to two of the influencers. And we're going to talk about the Locus Coeruleus, which is a brainstem nuclei. It's our noradrenergic hub. And then we're also going to talk about the nucleus basalis of minor. I welcome questions. And then can you remind me, we end right at the top of the hour, correct? Okay, great. So the main clues we'll talk about are the ones that I had to go to the literature to really appreciate. And so we'll look into these from the glial disturbance perspective and hyperactivation of neurons. But there is a plot twist that we'll get into. And part of that will be the contribution of the Locus Coeruleus vulnerability to a leaky blood-brain barrier that we hypothesized to really be impactful in this plasma p-tau release, and the relevance to cognitive decline. And so why do A beta measures in the brain correlate so strongly with plasma p-tau levels was our challenge. And I'm very fortunate to be part of a large study that was spearheaded by Prashanthi Vamuri, John Graf Bradford, and Michelle Milkey at our Mayo Clinic, Minnesota campus. But I really kind of became interested in this mystery when I was a part of a lecture series where they described CSF total tau increasing in these APP presenilin mice models. And at first, I really did do a double take. This is supposed to be a mouse model that's very specific to amyloid, doesn't have a tau component. So why would we see this total tau increasing? And one of the reasons for my double take is in the field of Alzheimer's disease, unfortunately, people either have studied tau or they study A beta, and almost to the exclusion of others. And I've witnessed this over the years. And it's really, we study a disease that has two pathologies that define it. And so I wanted to make sure that this was not sort of an agenda being pushed. Data doesn't push its own agenda, but I did do a double take, not in the work that they did. It was very strong work, but in this connection, such a strong connection between the A beta measure and the plasma p-tau. And one of the reasons that I paid attention so much to this is in the past, when we were looking at amyloid beta plaque patterns in the context of the amyloid PET imaging, we built out a really nice regression model that sought to predict cognitive decline using MMSE. This was the relative cognitive measure that was available in our cohort. And this is a score from 0 to 30, high being good. And what we found is when we modeled this information, so we put all of these different variables into the model, sort of predictive variables, what predicts the cognitive decline? We found that it was Brock stage, so it was the tau component that would predict. So for every single stage higher, you could actually have 5 points lower, which is quite impressive. But in this instance, we didn't see amyloid phase, the thal phase, overly predicting the decline. And so I've often paid attention to correlations where people are looking for A equals B, but correlation doesn't equal causation. We always have to be thinking about other players in mind. And so I was really fascinated by this study to understand what was this connection. And so wanted to take the opportunity to talk a little bit about perhaps there was a cortical detection threshold. These cliff Jack curves that are really quite popular at conferences are very helpful in understanding this hypothetical curve of when biomarkers become available. But what I'd like to make sure to highlight is the amyloid PET that precedes the tau PET. This is specific to the cortex. And so whenever we're looking at data, we really want to understand, is the detection threshold or what we're looking at, does it sort of inversely relate to another? Tau PET, so when we look at tau pathology, it first develops in our entorhinal cortex before going on to limbic cortices. And that's in contrast to amyloid, which begins on the outer part of our cortices, before then going into diencephalon and limbic regions. And then also, you can see this nice inverse relationship. So it's basically a bottom up for the tau pathology and a top down for the amyloid pathology. And so really kind of keeping this in mind of these sort of differences. And it's okay for two pathologies not to align well, but it's the two pathologies together that defines Alzheimer's disease. And one area that's often overlooked, but increasingly is of interest to many groups, is the locus coeruleus. And Brock and Kelly Del Tredici introduced this concept that the tau pathology may actually first be occurring in our brainstem before involving other structures. So I've kept this in mind, and especially in the context that it's actually at the very end of thal phase when we start to really see some of this brainstem involvement. So is it a cortical detection threshold, or is there some type of threshold that was allowing for this A beta and the plasma p-tau to relate so well? Or could it be something called tangle maturity? So on the right-hand side is a beautiful image from Christina Maloney's recent study. And so she had a beautiful review, and then her recent study where she characterized each of the tau antibodies that are very common in fluid biomarkers across an ever-expanding tangle lifespan. So when a neuron starts to accumulate tau pathology, it accumulates it as a pretangle before involving it to more of a fibrillar form in the mature tangle, and then eventually killing the neuron in the ghost tangle form. And we see a great deal of recognition of these plasma markers in the earlier parts of tangle maturity, but this precipitously drops off in the more advanced. And we think that this may underlie why the fluid biomarkers are so readily observed prior to tau PET and prior to some of the tangle accumulation that's being marked. And so that was really in the back of our minds as we were approaching this thought. But how do you actually approach the mystery? What can we actually look at, and what can we provide to the field to be able to better understand some of the differences? Well, one area and one of the mechanisms that is very important when translating your information is to put it into a global scale perspective, something that other groups use. Everybody uses the ABC, amyloid, Brock, SMADRC, so the thal phase, the Brock stage. We wanted to understand it more in the context that others would understand. And then we also wanted to use our expertise in digital pathology. And so we focused in on A beta measures, but we also investigated both the phosphorylated form at threonine 181 and 217. So these are the two plasma measures that we had access to. We looked at it in the brain to be able to understand some of the differences. Throughout the paper and throughout this lecture, I just want to highlight that when I talk about p-tau, I'm referencing the plasma biomarker levels. And then the PT, the phosphorylated threonine form, this is in direct reference to the digital pathology, to those antibody-based methods. And then with regard to who are we going to actually get to study, we evaluated our population-based Mayo Clinic study of aging who had plasma tau measures, this p-tau measure, and it was available within the 3 years. And the 3 years is something that I recommend to individuals that are performing clinical path or radiologic neuropath, because we want a tighter window if we're wanting to understand those associations. And this resulted in 35 autopsy participants. And I'm very fortunate to be surrounded by wonderful coordinators, a wonderful IHG team, and several very strong fellows that participated in this study, because it was a large endeavor. 35 doesn't sound like a lot, but when you start to think about all the slides stained, all the blocks that needed to be viewed prior to slide staining, and then the digital pathology analysis. It definitely took a village to get this paper published. And so now expanding a little bit about how we would plan to approach this mystery. When it comes to human data, pretty much always we can run normality tests, but I recommend non-parametric statistical tests so that you take away some of the assumptions. By enabling the non-parametric, you can look at rank-based correlations. I love regression models because it helps me to understand the contribution of variability. As human beings, we are quite variable, but we can actually look to see what's contributing to some of that variability that we have access to. And so this does help with the smaller sample size, but actually, quite frankly, my brain loves patterns. And so if I can understand the pattern, especially from a quantitative perspective, it helps when trying to solve said mystery. Now, we did have some confounders, and we wanted to understand them. In this instance, I don't necessarily like to adjust out for confounders. Instead, I like to plot the data and understand it in the context for which we'll be evaluating. And then what do you do with 35 if you have multiple different diseases? Well, you can actually take advantage of their bins, and you can visually inspect the changes. We don't have the power to be able to look at group-wise differences, but we can inspect the graphs to appreciate what an Alzheimer's case may look like. And in comparison to cases that had less than a Brock stage, was there significant amyloid pathology or was there relatively insignificant amyloid pathology? And then what's actually happening in our primary tauopathies? The previous literature showed striking lack of contribution to plasma p-tau levels in the primary tauopathies, and we wanted to kind of get a sense to see this in our own hands. So we had learned that the kidney health and liver health may be affected in or can be affected in Alzheimer's disease. But more so, what we observed and confirmed previous studies was that the kidney health specifically, and not necessarily liver health, needs to really be examined. I don't think that we're going to have to throw out cases that have increasing kidney maybe deficiencies, but chronic kidney disease does appear to contribute significantly to a much higher plasma p-tau level. And so we didn't see differences with regard to liver health. And then when we looked at age and time from plasma draw, we did see the same case that seemed to contribute, but not from the perspective of age or time. And so we have not adjusted for that in the analyses. But this specific case was removed to avoid undue biasing of our data. And once again, showed up further on. So with that case removed, we have the N of 34, and you guys can see now that we've visually plotted the different types of disease states. But really, what I was struck by is how strong the correlation was both by Thal phase, so our global amyloid measure, and both p-tau 181 and p-tau 217. And so we're talking about correlation coefficients of 0.56 and 0.71. It's really high in the human system. Now, in comparison, I thought the digital pathology would have provided even stronger correlations. And while it wasn't as strong as the global measure, we still see a highly significant correlation that enables us to really, in our hands, demonstrate this A beta in the brain, so the sort of deposition, in comparison to this plasma p-tau level. Now, on the bottom, what I've just gone through is both the hippocampus and parietal. And what I'd like to highlight is the hippocampus associates to a lesser extent than the parietal cortex. And we actually do see really strong and very similar correlation coefficients when we look at a cortical measure, and more so than the limbic measure. And so now segueing over to our tau correlates, what we can see is we do similarly see a strong correlation, but not as strong as with the A beta, which I was very struck by. With regard to our Brock stage, we actually only saw a trend with the hippocampal data, but not significant differences until we looked at parietal cortex. And this is the theme, is that we really see a much stronger relationship with the cortical measures of either A beta or the p-tau when we measured it with the PT181 antibody or the PT217. And so just to remind you, this is data that we were able to obtain from the brains, the immunohistochemical measures. What you see here in red is the positive staining in comparison to the soluble fluid tau levels. Some of the cases, 7 of them actually did have tau PET, and we were interested to see that the tau PET uptake in the parietal was able to show a really strong correlation on par with what we were observing. So 0.53 and 0.71 in both the 181 and that's supposed to say 217. So pretty impressive there. But as I told you earlier, I really like regression models. I really do enjoy looking at associations, but I want to keep in mind what may be contributing. And so we'll work toward those, but what I want to show you here is that we did similarly see a nice relationship that we saw with thal phase, but also if we just specifically looked at diffuse plaque score or neuritic plaque score, the much stronger relationship with the PT181, so the tau burden in the parietal cortex. So whenever I approach a regression analysis, I first look at what I've just observed from the association standpoint, and I'll then bring the stronger relationship into the model so that I'm not over modeling the data. And so in this instance, we're going to be looking at a Brock stage, a thal phase model, so more of a global model, and then those that have our digital pathology. But there's a lot going on in the Alzheimer's disease brain. When we look under the microscope, it's not just plaques and tangles. We can see TDP-43 pathology that can be classified using the late NC, so amygdala, hippocampus, frontal, so a staging scheme. We can also look at cerebrovascular disease. There aren't that many established neuropath scales, so if anybody in the audience has one they'd like to recommend, we love trying to apply them in our own data. But Raj Kalaria's group had put out recommendations for a scale that we were able to apply, and that gave us a score from 0 to 10, 10 being worse performance. And that information was built in. And so on the left-hand side, what I'm showing you is our p-tau 181, so our fluid p-tau 181 and our fluid p-tau 217 models. And into the model, we've included Brock stage, thal phase, the TDP-43, and the cerebrovascular disease. And impressively, we account for 31% of the variability, but it is actually the amyloid that continues to drive the relationship, which I was struck by. But this is why we set out goals, we set out to test hypotheses, and it is very okay when the biology goes against what you would have necessarily predicted. But what I want to highlight is in the MSD p-tau 217, we do see both Brock stage and thal phase significantly predicting the actual increase in the fluid marker. And this accounted for a really impressive 59% of the variability. And we saw a similar trend with the digital pathology data, just not as strong. None of the predictors remained significant for the p-tau 181, but in the p-tau 217, it was amyloid that remained the significant predictor of p-tau 217 in the fluids, which was very interesting. Okay, so this is our intermission, keeping our sort of mystery theme. And so our data suggests that tangle pathology may still play a supportive role, but that A beta may be playing the starring role, if you will. And our background characters are TDP-43 and cerebrovascular disease. We didn't see a significant contribution. I do want to still try out other types of cerebrovascular disease scales, but in this context, we've only seen either A beta or A beta and tau contributing. But what role do the influencers play? So if we still take a larger step back and we focused on our Brock tangle stage and our thal amyloid phase, but we do have 2 major cholinergic hubs that can impact Alzheimer's disease neuropathology. And so we and others have previously shown really the strong impact of nucleus basalis of Minor as that neuronal loss declines, or as the neuronal loss occurs from the tangle accumulation, you can see an effect of the cholinergic projections throughout the brain. And this has more of a direct relationship with tau pathology. the Locus Coeruleus, the noradrenergic hub, has had a lot of really interesting studies that have demonstrated that its effect on glial pathology, but really a nice relationship with amyloid pathology in the cortex. And so in order to systematically evaluate the contribution of either of these in a larger series, we came up with a way to neuroanatomically assess the levels. And so I'm going to talk to you today about data that is derived from the anterior level. And this is really work that is inspired by Mesulem and Guela and a lot of their tracing studies in non-human primates, and really helps to capitalize on an ideal section to evaluate. Interestingly, in Alzheimer's, the effect is much more prevalent in the posterior portion of the nucleus basalis minor and then eventually anterior, but it's so burnt out at end stage, and we don't actually relatively capture that region, that the anterior they recommended was nicely organized nuclei. So we were able to actually quantify through tracing and through creation of digital pathology algorithms. And then the Locus Coeruleus is really helpful in that we also, from a rostral-caudal extent, could define, but also nicely quantify the number of neurons present. And so on the right-hand side, I'm showing you images that Christina Maloney designed using a machine learning component that has pattern recognition, where she actually suppresses the neuropil to prevent the algorithm from misclassifying vessels or what have you. And so it really nicely captures the number of neurons. Okay, so we've talked about neurotransmitter hub involvement, but why? It seems a bit tangential. Well, the thought was, if nucleus basalis of minor is supposed to associate so strongly with tau pathology, and Locus Coeruleus associating so strongly with amyloid pathology in the cortex, it stands to reason, based upon our prior findings, that the Locus Coeruleus would have a stronger association with the plasma levels than the nucleus basalis of minor. And in point of fact, this is what we found. So the nucleus basalis minor is on top. We don't see a significant correlation. With regard to the Locus Coeruleus, though, we do see a nice relationship inverse, such that as plasma p-tau increases, there are fewer and fewer neurons. So suggesting that Locus Coeruleus vulnerability associates with plasma p-tau. There could, of course, still be some intermediaries in between, and we didn't have enough power in these models to actually model it. So we're very excited to extend this finding. Okay, but what about cognition? This is what we all think about. What is the actual impact? So the lovely part about plasma p-tau or fluid biomarkers in general is they give us this window of opportunity to look early in the human condition. But cognition is this very strong outcome measure because it allows us to understand what is the impact on our patients and our study participants. And I'm just reminding you of our earlier model that I talked about, where we saw that Braak stage significantly predicted MMSE score, and such that for every increase in a Braak stage, we saw 5 points less, and that tau amyloid phase not. So we focused in this instance on the insoluble forms of the disease and not necessarily on soluble levels. So we're going to switch around the model just a little bit to be able to predict cognition as the outcome. And so similar to what we saw before, we have our variables listed. And on the right-hand side, once again, we have PT181, the p-tau, 217, 181, 217. And so we focus both on CDR sum of boxes, and this is also a high is worse. And MMSE, in this instance, the high is actually good. And so the reverse of the signs. When we looked at the CDR, so the Clinical Dementia Rating Scale, we found for every one Braak stage higher, 1 point was increased in CDR. And we found that it was actually Braak stage and not the fluid p-tau 181 that significantly predicted cognition when you accounted for them all together. And when we looked at the MMSE score, we similarly found that it was Braak stage, so that insoluble accumulation of tau pathology in the neurons was significantly predicting cognition. In this instance, for every one Brock stage higher, we saw almost a point lower on MMSE when accounting for all of these. And a part of this is we are including an early biomarker in there. So I wonder if that's some of that muted effect. And we saw this similarly for both 181 and 217. But the amount of variability was not as large as we had observed earlier, but still pretty decent. 25% variability in a human condition, or 30 and 32% is still capturing quite a lot when there's so many things that could contribute to cognitive decline. And so I want to take us now toward and back to what we started at in the beginning, talking about what we propose as this intersection hypothesis. And so our mystery really was, why does A beta that accumulates in the brain associate so strongly with plasma p-tau, the soluble p-tau release? Well, I want to focus in that we propose this in the cortex. So these are cortical observations. There was a really great study from Oscar Hansen's group where they also similarly found cortical associations with the 217 measure. And so what we'd like to talk about, and what I'm going to work toward, is proposing parallel processes. To many people in the audience, this is really straightforward, but if you only focus on one or the other, and this started to even happen to me, I'd focus so much on tau pathology. And so I very much welcomed A beta experts into the lab so that we can continue to talk about both of these processes in parallel. So we're going to talk about the plaque-mediated route and the tangle-mediated route. But I'd like to first talk about the locus coeruleus vulnerability in this proposal, and what that might be doing toward and what has been proposed as far as glial disturbance. So there's some really cool work in the literature where they ablated the locus coeruleus, and they found that when they ablate the locus coeruleus, the ability for the microglia to really phagocytose and engulf is impacted in this condition. And so some of the thought here is locus coeruleus vulnerability in the context of Alzheimer's disease may be affecting the glial reactivity in the cortex that could potentially be influencing the A beta accumulation, so enabling a lot of the A beta accumulation to continue. Another really interesting study, or sets of studies that help to kind of connect the dots, is the work that's been done looking at the hyperactivation of neurons in the context of A beta plaque development. And so what this shows is it circles out A beta plaques, and what it found was that the hyperactive cells were much more common as they were closer, or if the plaque pathology was close to a neuron, it was more likely to have been a hyperactive cell. And so this idea that perhaps the accumulation of the A beta in our parenchyma may be causing a hyperactivated component. Okay, so why does this matter, you may ask? Well, the plot thickens. As we can look further into the literature, there's really interesting observations that if you hyperactivate a neuron, so this is outside the context of amyloid, but if you hyperactivate a neuron, there was more soluble p-tau that was observed in the mouse models that they were affecting. And so although these two studies are not connected, we started to really connect the dots that perhaps this locus coeruleus involvement that's then affecting the glial reactivity of our amyloid beta plaques was enabling sort of this, or the induction of hyperactivation, which could be influencing the neuron to release soluble tau. Now, I don't know how many of you guys are familiar with the studies, but there are studies in hibernating bears. So there's many conditions where we actually do accumulate phosphorylated tau. I mean, the point of tau is to be phosphorylated to allow for that flexibility. It's the hyperphosphorylated state that makes it a lot more difficult. And in this context, it's fascinating to think, what if there is this homeostatic imbalancing that occurs as the release of soluble tau during the same time, potentially, of accumulating phospho-tau for other reasons? Now we're going to go in the parallel. Okay, so we've talked for a long time about this plaque mediated. What about the tangle mediated? When we look at studies that we and others have designed, we can see this really strong association, especially if you look at this across Alzheimer's disease subtypes, where we have variability of brain regions that are affected. And what we see, especially in our younger individuals, is a much greater involvement of the nucleus basalis of minor. And so as the nucleus basalis of minor accumulates tangles, unfortunately, the neurons are dying and then having this effect on projection and causing what we propose and others, neuronal dysfunction in the cortex. But this neuronal dysfunction, while it may be having an impact on tau accumulation, this tangle maturity, there still is this feedback as neuronal injury. In the end, the tangle pathology, what we see, it's a stress condition. And so this neuronal injury may then be providing more information to the neuron that could be impacting. And so that could be some of the tangle contribution to the soluble p-tau, but it still seems really unimpressive in the context of the A-beta. But if we start to think about sort of some plot twists and talking again about the locus coeruleus vulnerability, once again, there's other really interesting lesioning studies that find that in the lesioned animal, there is a blood-brain barrier compromisation. And so this microvascular permeability that can be shown here with albumin could potentially be the reason that as the neuron's releasing soluble tau, we may be able to see even more as the disease progresses because of the leaky blood-brain barrier. The other plot twist being that we want to think about cognition really in the context that the direct route on cognitive impact may actually be going through somewhat of a circuitous path and not necessarily the direct accumulation of A-beta plaques, but instead perhaps this feedback from the neuronal injury, providing feedback and potentially conferring more of a homeostatic imbalance. Okay, and so I wanted to give us time to conversate. I can also keep in mind that it's bright and early in the morning and people haven't had their coffee, but I would be remiss if I didn't thank my incredible team and my funding and my collaborators. This has been a really rewarding experience to venture out into an area that I was not comfortable with and not familiar with, but I think along the way, we've very much enjoyed the process. So without further ado, thank you for your attention.